Hello everyone, welcome to EPG Pathshala. I am Dr. Veda Krishnan. I am a scientist in the Division of Biochemistry, Indian Agriculture Research Institute, IARA, New Delhi, India. I am going to talk about a very relevant topic, Purification of Enzymes, Part 2, from the paper Protein Biochemistry and Enzymology. In this lecture, the focus will majorly on understanding the basic technique of column chromatography, which is used for protein and enzyme purification. This will be followed by a brief description on different chromatographic methods that are used for protein purification, in which the basis and principle of each of these methods and advantages and limitation will be discussed. To start, this concept map shows the outline of today's lecture. In this, we will first briefly study about the properties based on which protein separation is achieved. Next, an understanding of basic chromatographic setup and equipment and the components required, and at last, on different chromatographic purification procedure, namely size exclusion, ion exchange, affinity, hydrophobic interaction, as well as high pressure liquid chromatography. Proteins can be separated on the basis of solubility, size, charge, binding affinity, and hydrophobicity. Usually, protein mixtures are subjected to a series of separation, each based on a different property to yield a pure protein. Chromatographic separation of protein mixture are among the most effective and widely used means of purifying individual protein. A variety of chromatographic purification techniques are available. Some of the most commonly used method on the basis of the property on which separation is carried out is shown in this table. The basic components required for column chromatography as shown in this figure. So, what is a column? A columnar glass reservoir of various length and diameter, which is cleaned, pre-packed with a matrix or resin and is attached to a chromatographic setup. It is normally a stationary phase, consists of an inert matrix, which is a resin, which is packed into this column having functional group bound to it. Mobile phase or solvents are another part which is constituted by various buffers and used for column washing, equilibration and elution of the bound protein from the stationary phase. Next is the protein sample. The protein sample to be purified is an essential component which should be present in sufficient concentration and loaded onto the chromatography column. In this figure, you can see a laboratory setup of protein purification. Let us discuss about each parts. A pump. A peristaltic pump with variable and adjustable flow rate is used to push buffer through the column. Fraction collector. This is necessary for collecting the fraction as when they are eluted out of the column. A detector. The fraction collector is usually connected to a detector which record the protein absorbance at 280 nanometer. Fraction can also be read individually on a UV spectrophotometer. Standard assay to measure protein content and activity is another important parameter. A standard assay for determining the protein activity and amount of specific protein eluted is very necessary prior to any downstream processes. Column chromatography can be performed with automated system using a pump to force the solvent over a packed column at a set flow rate, or it can be run under gravity. Both automated and gravity flow system can be coupled to automatic fraction collecting system. There are advantages and disadvantages to each system, which is shown in table 2 in the next slide. Column chromatography can be performed with automated system wherein a pump is used to pass solvent over a packed column as described earlier, or can be run by gravity flow, in which the solvent run down the column due to the force of gravity. This table enumerates the major advantages and disadvantages to each system. it is evident that automated methods are advantageous over gravity flow method in terms of ease of operation, programmability, ease of setting up a gradient elution and reproducibility of result being produced. Contrarily, the need for costlier purification equipment, chances of column black flow are the issues of concern. While gravity controlled methods are less expensive and give greater user control, it is more labor intensive and time consuming. Also, the results produced in repeated experiment are not easily reproducible. The steps for preparing resin are shown in this figure. Resins for protein purification are available in dry or pre-swollen form. In case dry, the resin need to be hydrated before it is used for column packing. This is done by mixing the dry resin with buffer and leaving it for overnight incubation at room temperature. After the hydrated resin has settled, very fine particles which remain suspended at the top are carefully decanted and discarded. The next step is the equilibration of the hydrated resin in the purification buffer. Finally, the slurry is degassed to minimize the bubbles prior to packing of the column. There are a number of chromatographic methods that are in use for purification purposes. The major type of column chromatography include size exclusion chromatography, ion exchange chromatography, affinity chromatography, immobilized metal ion chromatography, hydrophobic interaction chromatography and high pressure liquid chromatography. Usually, a protocol for protein purification may contain one or more chromatographic steps. It is essential to choose the most appropriate chromatographic method for optimized protein purification. The figure shows the separation of protein on the basis of size of the protein molecule, or known as size exclusion or gel filtration chromatography. The stationary phase comprises of porous bead made of polymers such as dextran or agarose or polyacrylamide, which are both insoluble and highly hydrated. Example of such commonly used beads which are commercially available are Cefadex, Cefarose, Biogel and many more. Extent of cross-linking can be controlled to determine the pore size. When protein mixture sample is applied to such a column, small molecules are able to diffuse into the internal volume of the bead and are thus distributed in the aqueous solution both inside the beads. On the contrary, large protein molecules are unable to enter the beads and are located only in the solution between the beads, as you can see in this figure. When the mobile phase, that is a buffer, is passed through a column of this material, molecules are fractionized on the basis of their sizes and shape. The large molecule flow more rapidly through this column and emerge first because a smaller volume is accessible to them. Molecules that are of intermediate size will occasionally enter and exit from the bead. These will thus flow from the column at an intermediate position, and small molecules which take a longer tortuous path will exit last, which can be clearly seen in this figure. Gel filtration chromatography is also known as molecular sieve chromatography or gel permeation chromatography. What are the advantages of gel filtration chromatography? Separation process is carried out under very mild condition. High resolution can be achieved by this method. Compatibility with many solvents is a positive attribute, thus wide range of buffer system can be used. Gel filtration can be used for buffer exchange, desalting or to obtain an estimate of molecular size. Coming to the disadvantages of this chromatography, it is very sensitive with the column packing. Column packing should be done ensuring there are no crack or air bubble trapped in between. Non-specific interaction between the protein and the resin can often lead to the decrease in resolution. So dear friends, the next method is ion exchange chromatography. Ion exchange chromatography or IEC is the most frequently used chromatographic technique for separation of proteins, peptides or other charged biomolecules. The basis for separation of protein in ion exchange chromatography is the net charge on the protein and their ionic interaction with the matrix that comprise the stationary phase of the chromatographic column. The stationary phase comprises of a column of bead containing carboxylate group which act as a cation exchanger, and positively charged protein which are cationic protein will readily bind onto this cationic exchange column. For example, carboxymethylcellulase column due to this electrostatic attraction. Similarly, negatively charged protein can be separated on positively charged diethyl aminoethyl cellulose, that is DEAE cellulose column, which is an anion exchanger. Separation of positively and negatively charged protein on an anion exchange matrix is shown in this figure. A common strategy for elution of such a bound protein is to use an increased salt gradient, that is, to increase the concentration of a salt, for example NaCl, in the eluting buffer. The sodium ion will compete with the positively charged group on the protein for binding to the resin. Weakly charged protein get eluted at lower salt concentration, while protein with the higher charge density get eluted at high salt concentration. Look at this figure. Gradient elution also lead to increased resolution. Coming to the advantage of IEC, elution takes place under milder condition, thus enabling the protein to retain native conformation throughout the process. High protein binding capacity, and the method is easily controllable. Ion exchange resins can be reused for a number of cycles with appropriate column washing and sanitization. Affinity chromatography exploits the high affinity of many proteins for their respective ligand. The figure outlines the basic principle of affinity purification. As a protein mixture is passed through a column in which a ligand is attached to a matrix, proteins which have specific affinity for the ligand bind to the column, while other proteins pass out as unbound in the flow through. The column is then washed with buffer to remove all unbound protein. The elution of the bound protein is achieved by passing a high concentration of the ligand through the column or by altering chromatography condition such that of the binding affinity is decreased. A well-known example of affinity chromatography is lectin affinity chromatography. In this case, lectin protein concanavaline A, which has specific affinity to glucose, is purified by passing the crude extract through a column containing covalently attached glucose residues. Concanavaline A will bind to the column owing to its strong affinity to glucose. The bound concanavaline A can then be eluted out the column by passing a concentrated solution of glucose through the column. IMAC is a kind of affinity chromatography which is based on covalent binding between the protein, specifically the histidine residue in the protein and metal ion. In this process, the mixture of the protein can be separated based on their differential ability to interact with the metal ion. The technique is mainly used to purify recombinant protein since naturally occurring protein do not usually have an affinity for metal ion. Proteins are engineered to contain a polyhistidine tail such that histidine can act as a ligand for the divalent metal cation. The stationary phase is simultaneously immobilized with divalent metal cation. As the his-tag protein is passed through the IMAC column, those proteins containing a high number of histidine residue would be able to bind to the column more tightly than those with fewer histidine residue. The most widely preferred method of IMAC is to use an immobilized nickel column, which is known as nickel NTA, nitrilo triacetic acid resin column, or nickel IDA, that is nickel iminodiacetic acid resin column. The histidine residues of recombinant protein of interest bind onto this column. The elution of the bounded protein can be achieved by three different elution strategies: namely competitive elution, stripping elution and pH adjustment. Hydrophobic interaction chromatography or HIC is another powerful method of protein purification in which the protein gets separated on the basis of the relative hydrophobicity. HIC media are comprised of porous and cross-linked cellulose particle to which a hydrophobic group covalently bonded via a short spacer. For example, the hydrophobic group commonly used are butyl, phenyl or octyl group. For example, phenyl sapphirose matrix has phenyl group attached onto agarose support matrix. Protein sample to be purified is applied to an HIC column under high salt buffer. The salt in buffer reduces solvation of protein. As solvation decreases, the hydrophobic patches on the protein surface are exposed, which get adsorbed into the hydrophobic area on the solid support. Please look into this figure. Proteins bound onto HIC column are eluted under low salt concentration. Decreasing the concentration of salt reduces hydrophobic interaction, resulting in the protein getting desorbed from the solid support. So what are the advantages? Fast rapid chromatography due to high flow rate, can be easily operated on a large scale. The column does not shrink at high salt concentration and a wide range of solvent system can be used. An advanced and improved version of column chromatographic technique discussed so far above is the high pressure liquid chromatography or HPLC. Use of this method substantially improve the resolving power because the column matrix material are much more finely divided, enabling more interaction site and increased separation among protein is achieved. Since the column is made of finely divided materials of few micron and the pressure is required to be applied onto the column for obtaining adequate flow rate and good separation. The schematic overview of various steps in HPLC can be shown in this figure. Dear students, in this module we have understood that proteins can be separated on the basis of solubility, size, charge, hydrophobicity and binding affinity. Column chromatography is a widely used approach which is based on the principle of separation of a large pool of protein into many small pools, among which some may contain the protein of your interest. The basic components of a column chromatographic setup include a chromatographic column, a pump, a fraction collector and a detector, along with resin, protein sample and buffer systems. A number of chromatographic methods that are in use for purification purposes are size exclusion chromatography, ion exchange chromatography, affinity chromatography, IMAC, HIC, HPLC and have been studied all in detail in this tutorial. Thus, in nutshell, in this lecture we have learned about the chromatographic procedures involved in protein purification, an overview on the basic equipment and setup of chromatography. Along with this, the principle, the advantages and disadvantages. Thank you.